The human genome, a vast repository of genetic information, holds the blueprints for life. Understanding this intricate code is crucial for advancing biological knowledge and medical applications. Within this code lie motifs and domains, recurring patterns and structural units within proteins, which offer crucial insights into gene function and evolution. Motif analysis focuses on short, conserved sequences that often play roles in DNA binding or protein interactions. Domain analysis, on the other hand, examines larger, independently folding units of proteins, each with a specific function. These analyses are powerful tools in genomics research, enabling scientists to decode the language of life. They provide a framework for understanding the complex interplay of genes and proteins that drive biological processes. By identifying and characterizing these recurring elements, researchers can gain a deeper understanding of how genes function, how they interact, and how they have evolved over time. This knowledge is fundamental to advancing our understanding of health and disease. Motifs, like subtle musical phrases, contribute to the overall melody of protein function. They can be binding sites for other molecules, signaling sequences, or sites of post-translational modification. Domains, like distinct movements in a symphony, represent larger functional units. They can have catalytic activity, structural roles, or mediate interactions with other proteins. Together, motifs and domains provide a rich tapestry of information about a protein's role within the cell. These structural elements can be likened to the individual components of a complex machine. Each motif and domain contributes to the overall function of the protein, much like gears, levers, and springs work together in a clock. By studying these individual components, researchers can gain insights into the workings of the entire machine, in this case, the intricate machinery of the cell. The study of motifs and domains is thus essential for deciphering the complex language of the genome. Gene families, groups of genes descended from a common ancestor, provide a fascinating window into evolutionary history. They often share similar functions, reflecting their shared ancestry. Analyzing gene families across different species allows researchers to trace the evolutionary paths of genes and understand how they have diversified over time. This comparative approach can reveal how gene function has adapted to changing environments and selective pressures. By comparing the motifs and domains within gene families, scientists can identify conserved features that are essential for function. These conserved elements provide clues about the core functions of the gene family and how these functions have been preserved across generations. Conversely, differences in motifs and domains can highlight regions that have undergone evolutionary change, leading to functional diversification within the family. Gene family analysis is akin to tracing the branches of a family tree. Just as family members share certain traits, genes within a family share common features. Studying these shared characteristics can reveal the history of the family and how different branches have evolved unique traits. Similarly, gene family analysis reveals the evolutionary history of genes and how they have diversified to perform specialized roles. The study of gene families is essential for understanding the evolution of biological processes. By comparing gene families across different species, researchers can gain insights into how complex biological systems have arisen and adapted over time. This knowledge is crucial for understanding the fundamental principles of life and for developing new strategies for treating diseases. Motifs and domains serve as powerful indicators of functional similarity between proteins. The presence of shared motifs or domains often suggests that proteins perform similar roles, even if their overall sequences have diverged significantly. This is because these structural elements are often crucial for the protein's function and are therefore conserved throughout evolution. Comparative analysis of motifs and domains across different species can illuminate evolutionary relationships. The presence of similar motifs or domains in proteins from distantly related species suggests a common ancestor. The degree of similarity can provide insights into the evolutionary distance between species and the timing of evolutionary events. This information is valuable for constructing phylogenetic trees and understanding the evolutionary history of life. By comparing the arrangement of motifs and domains within proteins, researchers can gain insights into the evolution of protein architecture. The shuffling and duplication of domains during evolution have led to the creation of new proteins with novel functions. Understanding these evolutionary processes is key to deciphering the complexity of biological systems. The presence of specific motifs and domains can also provide clues about a protein's subcellular localization and its interactions with other molecules. For example, certain motifs are known to target proteins to specific organelles within the cell, while others mediate interactions with DNA, RNA, or other proteins. This information is crucial for understanding the protein's role in cellular processes. Section 4. Applications in gene, categorization and function prediction. Motif and domain analysis are invaluable tools for categorizing genes and predicting their functions. By identifying the presence and arrangement of motifs and domains within a protein sequence, researchers can classify genes into families and predict their likely functions. This is particularly useful for newly discovered genes or genes with unknown functions. These analyses are essential for annotating genomes and building comprehensive databases of gene function. By systematically analyzing the motif and domain content of all genes within a genome, researchers can create a detailed catalogue of gene functions. This information is crucial for understanding the overall organization and function of the genome. The ability to predict gene function is particularly important in the era of high-throughput sequencing, where vast amounts of genomic data are being generated. Motif and domain analysis provide a rapid and efficient way to analyze these data and extract meaningful information about gene function. These techniques are also valuable for identifying potential drug targets. By identifying motifs and domains that are specific to disease-causing proteins, researchers can develop drugs that specifically target these proteins. This targeted approach can minimize side effects and improve the efficacy of treatments. Section 5. Contributions to drug discovery and disease research. Motif and domain analysis play a crucial role in drug discovery and disease research. By identifying motifs and domains that are associated with disease, researchers can gain insights into the molecular mechanisms underlying disease and identify potential drug targets. This knowledge is essential for developing new therapies and improving patient outcomes. These analyses are particularly valuable for understanding the genetic basis of complex diseases such as cancer and Alzheimer's disease. By identifying genetic variations in motifs and domains that are associated with increased disease risk, researchers can develop diagnostic tools and personalized treatments. Motif and domain analysis can also be used to study the evolution of drug resistance. By analysing the changes in motifs and domains that occur in drug-resistant pathogens, researchers can develop new strategies to overcome drug resistance and improve the effectiveness of existing treatments. The insights gained from motif and domain analysis are transforming the field of biomedical research. These powerful tools are providing a deeper understanding of the molecular basis of disease and paving the way for the development of more effective therapies. The continued development and application of these techniques will undoubtedly lead to significant advances in human health in the years to come.